Yep, I just want to say thank you for the organisers for inviting me here to speak today. Um, Before I kind of talk about some data, I just thought it'd be important to start defining the problem as to what we're actually trying to do with transmission stage drug discovery. So as Didier alluded to, the MMV's target candidate profile for transmission blocking suggests that it's these two properties here that are important: gametocytocidal activity, so activity against the gametocyte, and vector stage activity could also be desirable, but maybe not as important as gametocyte activity. So a transmission blocking drug must target the gametocyte, but could be useful if it targets the vector stage. But as always, there's extra layers of complexity to add to that. So gametocytes aren't a static population, they undergo development. So the development is generally thought of as in Plasmodium falciparum as stages 1 to 5, and over about a period of about 10 days, they develop and go through all the different stages. And around about stage 3, stage 4, they start to lose sensitivity to common antimalarial drugs. And by stage 5, they're pretty insensitive to most antimalarial drugs. And the problem with this is that it's the stage 5 gametocytes which are responsible for onward transmission. So the cells that are responsible for passing on malaria are the ones that are hardest to kill. And that's all well and good. It may be argued that hit the gametocytes early when they're young, when they are sensitive to antimalarial drugs, and you solve your problem. But unfortunately, again, it's not as simple as this. And frequently, when patients visit the clinic with malarial symptoms, they already have stage 5 gametocytes in their blood already. And in this particular study, approximately 80% of people had gametocytemia by PCR. So just recap there. We need to target gametocytes. We should probably prioritize targeting the stage 5 gametocytes, as these are the ones infectious to mosquitoes, and unfortunately they're drug sensitive. And we should target these cells because sick people already have them when they come for treatment. So it's not so good to treat them for the symptoms and then send them home again if they're already still infectious to mosquitoes. So also, I think it's important to think about the cell biology of transmission, as Didier also mentioned. So you have your gametocyte development from 1 to 5 in the human blood, and it's important to note that gametocytes are not a homogeneous population. There is both males and female gametocytes. And these stage 5 gametocytes are sitting in circulation waiting for uptake into a mosquito. And upon mosquito feeding, they recognize they're in the gut of the mosquito, and they suddenly and quite explosively differentiate into gametes, which fertilize each other, form an ookinete, which migrates through the midgut wall and forms an oocyst there, which eventually then develops sporozoites, which then go on to colonize the salivary gland and cause onward infection. So this is kind of the range of cell biology we're interested in. Now, probably everyone knows that the gold standard for looking at transmission at the moment is the Plasmodium falciparum standard membrane feeding assay. And this blue bar underneath illustrates the cell biology that this assay interrogates. And so basically you have these stage 5 gametocytes, you incubate them with your compound of interest, then you, for a period of time, 24 hours, 48, etc., then you feed them to mosquitoes and look about 7 to 10 days later at whether oocysts have formed. And if they haven't formed, you block transmission. And this is a very good assay, but unfortunately it's very, very expensive to do. There's very few labs in the world that are successful with this assay. You need insectaries to grow your mosquitoes. You need culturing facilities, and there's safety considerations. And it takes a very long time to perform the assay, and you need trained people to be able to interpret it. So it's not really that useful for very, very large-scale screening. So in this case, we have surrogate assays. And in the literature at the moment, generally these are kind of divided up into what are known as early-stage assays and late-stage gametocyte assays. And early-stage gametocyte assays, generally speaking, are looking at stage 1 and 2 gametocytes, sometimes stage 3 as well. And late-stage gametocyte assays generally talk about stage 4 and 5 gametocytes, and sometimes stage 3. And if you see here, the relative cell biology of an early-stage gametocyte assay has no overlap with the SMFA. So I think we can probably say that whilst important and interesting to know about early-stage compounds activity on early-stage gametocytes, it's not predictive of the SMFA. Late-stage gametocyte assays are much better because they have stage 5 gametocytes in them, which are also being looked at in the SMFA, but also they have this sort of slightly immature component as well, which might be a complicating factor to interpreting data. So we've tried to take a different approach. So we've developed an assay we're calling the dual gamete formation assay, the DGFA, in Plasmodium falciparum. And what we've tried to do is look at the cell biology of transmission and the SMFA and try and replicate as much of this in vitro as we possibly can. And currently, this part of cell biology in grey at the moment. So looking at the stage 5 gametocytes in the human host and then simulating mosquito feeding by adding mosquito factors to your culture to induce gamete formation. So this assay kind of came about by various appreciations that we've had over the last few years. And the first one of those is that mature gametocytes have finished their gametocyte development. They're sitting in the bloodstream, waiting to be taken up by mosquitoes. They're probably not doing very much, and they have sort of maybe minimal sort of metabolic activity to keep them alive, and probably try and evade the host that's trying to kill them. So to me, it seems that minimal activity suggests few drug targets. So it might be actually pretty hard to target these to kill them, and this is probably partly explains their drug insensitivity compared to earlier stages. But on the other hand, gametocytes are almost like coiled springs. They've got all of the machinery and mechanisms in place ready to undergo explosive development in the mosquito. And perhaps these are good targets because there's lots of things going on during gamete formation. And if we can target those in the gametocyte and break them somehow, maybe we can reduce their infectivity or prevent them infectious at all. We call this inhibiting the functional viability of the gametocyte, so its ability to function. And the second thing that we've appreciated is that gametocytes come in males and females, and the males are more drug sensitive than the females, but they're the lesser population, so there's approximately 5 times fewer males than females in culture. So you have this quite nicely drug sensitive population of males in your culture, but if they're a small population, unless you have a sex specific readout, you might miss any effects on those, and because it needs a male and a female gametocyte to transmit, you can kill one of the sexes and block transmission that way. So in the assay, it's a dual readout assay, so in one sample we can sequentially measure the effects of a compound on male gamete formation, and then further on measure effect on female gamete formation. So male gamete formation or exflagellation is measured by actually visual microscopy, by an automated microscopic system, looking at the vigorous beating of the male gametes as they emerge. And that happens probably about 20 minutes after gamete formation is induced. Female gametes are detected using a specific antibody which recognizes PFS25, which is a female-specific protein, and it's expressed on the surface of the female gamete, and we use live staining to show that our females are viable. And currently this assay can run in a 96 and a 384 well format. The Z factor of the assay in both formats is acceptable for screening, and as I said, it's a dual readout, so you get 2 readouts for the price of 1 with the assay. So one of our control compounds, methylene blue, has pretty much equal activity against male and female gametocytes. So the assay looks like it's working pretty well in our hands. So to take it further, we just recently published a proof of concept screen of the MMV malaria box, and I'm sure you all know this 400 compound library that's been selected by the MMV that all have activity against asexual parasites, and what we're doing is looking additionally, does it have activity in our gamete formation assay. And at 1 micromolar we screened all the library and we identified 18 compounds giving greater than 50% inhibition. And the assay conditions we used, we call this a carryover format of the assay. And basically what we do is we incubate our stage 5 gametocytes for 24 hours with the compounds of interest. Then we trigger gamete formation in the presence of the compound. So the compound is present throughout the whole process of the assay. And this allows us to identify a few things. So carryover, just here. Because the compound is present throughout, the compound could be targeting 3 different processes. It could be targeting the gametocyte, either by killing the gametocyte, so this gametocyticidal activity, or it could be sterilizing the gametocyte, so the gametocytes are still alive, but they cannot undergo any more development, so they've been inactivated. And this kind of matches quite nicely with the MMV's TCP. But in addition, because the compound is present during gamete formation, they can also target the process of gamete formation directly, and we call this a contraceptive effect. And this is vector stage activity, which it could be useful, but maybe not quite as useful as gametocyte targeted. So in our assay, in the carryover format, all three will give an inhibition, but we can't tell the difference between them. So to increase the information of our assay, we developed this washout format where it's exactly the same. So we incubate for 24 hours with our compound of interest against the gametocytes, but then we undergo some washing steps so that gamete formation occurs in the absence of the compound. And under these conditions, contraceptive compounds have no effect and gamete formation progresses, so we're kind of telling the difference between the gametocyte targeted and the gamete formation targeted compounds. And I just wanted to kind of jump to the end to cut a long story short. This is kind of our conclusions from our screen to date. And I hasten to add it's a small screen, and the MMV malaria box library is a biased compound library of asexual killing, so take everything with that in mind. But using our different formats, we can start to divide the compounds out and phenotype them. So we can identify male-specific compounds, we can identify female gametocyte-specific compounds, or dual-active compounds that kill both males and females. And we can look for irreversible effects, which is killing and sterilizing against gametocytes, or reversible effects, which is these contraceptive compounds. And the first thing I'd like to take away is that we don't identify a single female-specific compound. And I think we've tested probably about 1,000 compounds to date now, and we've never ever found a female-specific compound, which is interesting because they must surely exist, but I think this illustrates that male gametocytes are a better drug target than female gametocytes. We also try to characterize these compounds in a bit more detail by looking at publicly available data online, and we looked at the Chembl database, and we got cytotoxicity data against human cells. And we then evaluated this in comparison, IC50s in our assay compared to the CC50 in the cytotox assay. And anything that gave less than a five-fold difference in IC50 to CC50, we considered cytotoxic. And immediately you can see there's this group in the middle here, these dual-active compounds that are irreversible, and 4 out of 5 of them are cytotoxic. And this makes perfect sense. They target both male and female gametocytes. They're probably poisons, and actually these 4 compounds come out of pretty much every malaria box screen, regardless of which organism you're looking at. So probably they're just cytotoxic. Interestingly, and probably stands to reason, reversible compounds have pretty good parasite selectivity, and that's because if you remove them, the parasites recover and they can form gametes, so they're not killing anything. But up in this top left-hand corner, I think is particularly of an interest to me. These are male-specific compounds, so they only target the male gametocyte, and they're irreversible. So if you remove them before gamete formation is still inhibited. And these look like they have pretty good parasite selectivity. Well, they only target one parasite sex, so they're probably not killing everything. And I argue that probably these are sterilizing rather than killing. And so the sort of take-home message I have from this is that perhaps if we're looking for new transmission-blocking drug and starting points, perhaps this category here might be the most efficient way to accelerate drug discovery. And then just to revisit the validation of the assay, so it's important to validate an assay against current gold standards, the SMFA. So we selected 3 compounds with 3 different phenotypes. We selected a compound that is reversible and male-specific, a compound that is active against both males and female gametocytes and is irreversible, and a compound that's from this interest group that's male-specific and also irreversible. And we tried to ask whether these were the predictions in the DGFA held up in the falciparum membrane feeding assay. And to do this, we performed the SMFA in 2 separate formats similarly to the DGFA, so in this carryover and washout format, which is exactly the same protocol, but instead of triggering gamete formation, like in the DGFA, we just fed them to mosquitoes. And this was done with our collaborators in Roel Dinklesand's lab in Johns Hopkins. And quite gratifyingly, we found that our predictions were borne out pretty well. So when the compounds are carried over into the mosquito feeds, all compounds gave almost 100% inhibition at 1 micromolar. And then the compounds that we predicted to be irreversible, when we washed out the compounds before the feed, they still inhibited oocyst production. And then there was this one compound that we predicted to be 100% reversible when washing out in the DGFA. And whilst it didn't give 100% washout in the mosquito feed, it gave a rescue of about 25 to 30%. And I think this illustrates an important point. The DGFA does not include the whole of the cell biology of the SMFA, and probably this has downstream effects. And actually, since then, we've found that this compound has effects on urocinates, so there's a bit of an additive effect later on. So I'm pretty happy that our assay does actually have a good correlation with the SMFA, which is what we want for a screening assay. So just to conclude, I think it's really important that you think about cell biology of transmission blocking assays, because every assay can tell you something, but only if you know what it's showing you. And our DGFA is very cost effective. There's no purification steps or anything. It's just adding the cells to a plate, triggering gamete formation, which is not expensive, and then the only expense is the microscopes needed to analyze it, and that's a one-off cost. Now with the 384 well format, we can screen an estimated 25,000 compounds per month, and this is something we're going to be doing starting next year in collaboration with our colleagues at GSK. So I just wanted to say thank you for listening and giving me the opportunity, and thank you to all these people that have contributed to this work.
Michael, a really interesting essay. As you know, so the ATP4, you've done the pyrazole compound, which actually Didier, you did not box, which also actually does inhibit gametocyte, late gametocyte. So the malaria box has been screened by Kieran Kirk's lab and has found that there are at least 28 different compounds that seem to be affecting the sodium homeostasis, and so is the pyrazolamide compound that you tested as well. So I was sort of surprised that you had only 18 out of the 400 compounds that were actually doing this, and whether or not any one of these things were actually those that affected sodium homeostasis in Kieran's assay as well.
Well, that's very interesting. I mean, something I'm particularly interested in is actually trying to understand what all these compounds their targets are. So I don't know which ones are ATP4 inhibitors.
Oh, okay.
I'd be very interested to find out. But I would say that our assay conditions are quite stringent compared to maybe some other gametocyte assays. So we only incubate with our gametocytes for 24 hours. So I think we're probably, and screening at 1 micromolar is relatively low as well. So I think we're probably biasing towards the most potent compounds. And if we incubated for a longer time or a higher concentration, maybe we'd get a larger.
So Kieran screened all his compounds for at 1 micromolar, and that assay was actually just minutes. So that's the reason why I sort of there may be something else going on, which may be very interesting.
Okay, that sounds very interesting. I'll look that up.
It was done a very short period of infection. Yeah.
Time for one more question.
Thank you. Very nice work. Actually, this is more in a response to that. From the viability assay that we did with kind of stage 3 to 5 gametocytes, we also found that the malaria box was not very potent in any of those. So interesting to